Transformation of E. coli with Green Fluorescent Protein Plasmid by James Lomibao, Dominique Buckingham, Renato Soto, and Taylor Lloyd. Overview. E. coli was transformed using p-fluorescent green plasmid to express green fluorescent protein and ampicillin resistance. Genetic engineering is changing the genetic material of an organism so that it expresses different traits. One way that this could be done is through transformation. Transformation is when an organism takes up DNA from its surrounding environment and expresses its genetic information. This occurs naturally, or it could be done in the lab. For E. coli, it doesn't usually occur in nature. However, we were able to manipulate it so we were able to get that transformation. GFP was a gene of interest for this experiment. It's a sequence of DNA that encodes for green fluorescent protein. This protein absorbs blue light and expresses green pigment. This served as a marker to identify which were the bacteria or E. coli that were successful in transforming. A key tool for transformation is a plasmid. A plasmid is a small circular piece of DNA which typically contains an origin of replication, which is important for when the DNA is going to replicate. Promoter site, which allows the control or the manipulation of our gene of interest, which in this case was GFP. Restriction sites, which allow the scientists to integrate different genes into the plasmid, and a selective marker. In this experiment, it was ampicillin, which allows us to determine which E. coli were transformed and which were not.
For this experiment, we had 4 plates. Plates 1 and 2 were our control plates, where they did not receive the GFP DNA. Plate 1 contained only E. coli cells only, and without any more disturbing factors, we expected lots of growth. Plate 2 contained ampicillin with E. coli cells only. And the fact that they did not receive the GFP DNA, and therefore the gene that helps them that builds resistance to ampicillin, we expected no survivors. Plates 3 and 4 were our transformation plates that did receive the GFP DNA. Plate 3 contained ampicillin with E. coli cells and PFG DNA. Now, the fact that they did receive the PFG DNA meant that they were resistant to ampicillin, so there would be some growth. But the fact that they did not have IPTG, which helps inactivate the repressor for the promoter, there would be no growth. For plate 4, it contained ampicillin with E. coli cells, PFG DNA, and IPTG. So it did contain the GFP DNA that helps build resistance against ampicillin, so there would be some growth. And the fact that they did have the IPTG also means that it would also glow under UV light.
Procedure. 2 tubes were used for this experiment. One, labeled positive DNA, is the transformation tube. It contains the PFG plasmid. The second one is labeled negative DNA. This is the control. It does not contain the PFG plasmid. In each tube, ice-cold CaCl2 was added. E. coli cells were also added in each tube. Only in the positive DNA transformation tube, the plasmid was added. First, each tube was placed in ice for 10 minutes. They were then heat shocked in 42 degrees Celsius. This was done for 90 seconds and allowed the uptake of foreign DNA into the E. coli. This is where the CaCl2 importance comes in. When you increase the salt, it increases the stress. And increasing the stress is very important for the heat shock stage. After 90 seconds of heat shock, they were immediately transferred into ice incubation for 2 minutes. After 2 minutes, they were placed in recovery. Recovery. This allows the DNA to get settled. LB Luria broth was added to each tube. They were placed in 37 degrees Celsius. Luria broth was added again because it was important for the cells to recover. We will now discuss the plates that each tube will be placed in. There are 4 different transfer plates.
After placing the E. coli cells in their optimal temperature, they are then transferred on the plates. Plates 1 and 2 represented our control plates that did not receive the PGF DNA. Plate 1 contained E. coli cells only. Plate 2 contained ampicillin with E. coli cells only. Plates 3 and 4 represented the transformation plates that did receive the PFG DNA. Plates 3 contained ampicillin with E. coli cells and PFG DNA. Plate 4 contained ampicillin with E. coli cells, PFG DNA, and IPTG. After the plates were incubated overnight, they were analyzed. For plate 1, that did not have the PFG DNA and contained E. coli cells only, there was lots of growth. For plate 2, that did not contain the PFG DNA and had ampicillin with E. coli cells only, there were no survivors. For plate 3, that did contain the PFG DNA and E. coli cells, there was some growth but no glow. For plate 4, that did contain the PFG DNA and had E. coli cells and IPTG, there was growth and glow. The picture on the right represented the sample that did contain the IPTG and therefore shows some glow. The picture on the right was plate 3 that did not receive the IPTG and therefore doesn't have as much growth as the left picture. For transformation efficiency, we calculated that there was 200 transformants per micrograms. It can be concluded that transformation is not very efficient just based on the fact that out of the 4 plates in this experiment, there was only 2 that transformed.
All procedures were followed as indicated on the lab manual, Biotech Lab 6, transformation of E. coli with green fluorescent protein, plasmid, Bio 201.
Feed!